Friends, hi everyone. A very good evening to all of you. Very good afternoon. Hi everyone. Am I audible and visible to you all? Hello. Am I audible and visible to you all? Good afternoon. Good afternoon. Good afternoon, everyone. Am I audible, visible? Hello. Good afternoon. So how are you doing? Yes, everyone. How are you doing? Hi, Sunlight, Maya, Suhashini. What's up, everyone? So welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and today we are going to revise the anatomy of the flowering plant. You know that in the last session we have covered the tissue part, meristematic tissue, permanent tissue, and even we covered the entire tissue system, that how do we divide the tissue system. So in today's class we will start with the sections. Okay, about the root we will study, about the stem and about the leaf, and then we will talk about the secondary growth, which is a very important topic. Okay, so I am hoping, or I literally believe that my students today, they have prepared it well, they have prepared this particular chapter well, right? And here we all are quickly going to revise the concepts. Hai na? Hai na? So can I trust you today? So this is what I'm hoping, that today my students, they have revised this particular chapter and today they are here for the quick revision. So if I'm going to ask one question, then I'm going to get at the answer of that question. Hai na? And there will be the cross questioning as well. There will be the cross questioning as well. Okay, chaliye. So let's start the class, guys. If you're new to our channel, you know what you have to do. You just need to subscribe our channel and quickly hit the like button. Okay, so it's Sunday because you do not have your test today, and I think today you are busy and it's a long weekend. Na? See, Tuesday you have one holiday and it's just Monday then. So we can utilize this day, isn't it? Isn't it? So let's utilize it in a wonderful way. So everyone just hit the like button and share the video link with your other friends as well. And let's revise this particular topic, and mark my words, if you will complete both the sessions, right? If you complete both the sessions of this anatomy of flowering plant, right? Trust me, you will get all the questions from this part only. Okay, there in the NEET examination, you will not get anything beyond that. Fine. Okay, so now let's start the class, but before that, let me show you something. Can you see this here, this video? That 98% of the NEET aspirants, they do not know how to read the NCERT. Literally, it's a fact. We know that our paper is going to come from the NCERT, and we know that when it comes to biology, all the 90 questions will be there from the NCERT. But still, students are not able to score 360 out of 360, right? Sometimes 320, 330, and sometimes even below 300. So why is it so? If we know that paper is going to come from NCERT, then why we are not able to score good marks? So in this particular video, I have explained everything. So if you have watched it, very good. Otherwise, go and please check this particular video. And yes, I was checking the comments. So some students were saying that, ma'am, how to get full house or perfect score in biology. So here you people can check this particular video as well. So this is about the biology. Now when it comes to chemistry, you know that even chemistry is important, right? We need to score 180 marks there in chemistry, and it is also very easy. And you know what? And just to make it more easy for you, HSP sir has started a new batch. Right, he has started a new batch, and with all the mind maps he has started. So what you have to do, you just go to any video, right, bache, and you will find the description box here. There is the link for the HSP sir's batch. And I always used to mention it, bache, and I mean it. Students used to pay fees in lakhs to get admission in HSP sir's batch. And now you all are getting it just at a price of 1999. Okay, let me refresh this. Maybe there is some internet issue, that is why you're not finding this link properly. But otherwise, you know what you have to do? Just go and check the description box, right? Just go and check the description box. And what you have to do then you have to click on the link. Avenger coupon code is already applied, and you will get this badge at a price of 1999. And this batch is literally amazing. With all the mind maps he's going to teach you. And you know that when it comes to tricks, no one can beat HSP sir. So if you have joined it, very good. Otherwise, go and join it ASAP. I want to see you all there in HSP sir's batch. Okay. And do you want something like this for biology as well? Just funny things, just go to our channel, just go to the video section, you will find the solution of your problem. Yes, students, do you want something like this for the biology? Do you want something like this for the biology as well? Okay, ma'am, start something, some mind maps like things, or NCRT revision. Do you want something like that? Seriously? Chalo, I'll think of it and we'll start. So now quickly hit the like button and let's start this particular chapter, that is anatomy of flowering plants. Now, bache, the quick revision. When it is anatomy of flowering plants, when I have specified that we are talking about the flowering plants, then anatomy of flowering plant is the branch of botany where we are studying, right? It is the study of the internal structure. What is it? It is study of the internal structure. That is what anatomy is. And already, bache, I told you about it, that here in this particular chapter, how are we going to start? We are going to start with the tissue part first. So let me know in the chat section quickly, what is a tissue? Define tissue quickly in the chat section. Let's revise a bit and then directly I will start that sections wala part. Okay, so quickly tell me, what do you understand by the word tissue? Yes, guys, be quick. What do you understand by the word tissue? Exactly, it is a group of cells. You are right. What is a tissue? Tissue is a group of cells, and it's a group of cells having similar structure, similar function, and having the same origin, isn't it? It is the group of cell having similar structure, similar function, and the same origin. So what is the most important thing when we talk about the tissue? That the origin of cells should be same, right? That's the most important point, that the origin of cells should be same. So now when you are talking about the plants, we have already discussed that we have the meristematic tissue, which is actively dividing tissue, right? It will keep on dividing and it helps in the primary as well as the secondary growth of the plant, right? But this is what we have already covered. And second is the permanent tissue. So when you are talking about the permanent tissue, here cells are differentiated. What are cells here, bache Akhilesh? What are cells here? Cells are differentiated here. So if in one word I have to explain meristematic tissue, so it is the undifferentiated tissue. What is it, bache? It is the undifferentiated tissue. Here cells are just dividing, but here they do not have any permanent function. Okay, but when it is a permanent tissue, you know that cells are differentiated here. Now they have a permanent function. That is the meaning of permanent tissue here. Now when we are talking about the meristematic tissue, we have the primary meristem as well, we have the secondary meristem as well. And when it's primary, it helps in the primary growth of the plant. Basically it increases the length, and here it will increase the girth. Okay, the diameter. Clear, bache? Girth, the diameter. And when it is the permanent tissue, okay, when it is the permanent tissue, so here we talk about two things. One is the simple permanent tissue and another one is the complex permanent tissue. So bache, when it is simple permanent tissue, then you have your parenchyma, your collenchyma, your sclerenchyma. You have your parenchyma, collenchyma, sclerenchyma. Parenchyma, collenchyma, sclerenchyma. And when it is the complex permanent tissue, you have the xylem, you have the phloem, and they are going to form the vascular bundles. Xylem is meant for the transport of water and minerals. Phloem is meant for the transport of the food. So that's what we have already covered. Clear, bache? Clear, bache? And then what we discussed in the last class? See, it is a revision first, okay? So what we discussed in the last class, we talked about the tissue system. Yes, what we discussed there? We discussed the tissue system there. Fine. And when it is the tissue system, you know that you'll be having epidermal tissue system, ground tissue system, and the vascular tissue system. So if you have attended that class, then today's session is going to be very easy for you. So epidermal, ground, and vascular. Epidermal, ground, and vascular, that's what we have already covered in the last class. Clear, bache? Clear bache? So just for your quick revision, when it comes to the ground tissue, it is the largest tissue in the case of a plant, right bache? And here you will divide this ground tissue, especially in the case of the dicot stem. Okay, especially in the case of the dicot stem. Clear bache? Clear bache? Gopika, wish you a very happy birthday. Stay blessed, stay happy. Please focus here now. Okay, so tissue system mein epidermal, ground and vascular tissue system is there. Epidermal tissue system, ground tissue system, vascular tissue system is there. So this is what we have covered in the last class. So now what we need to start. See, if you have checked that session, very good. Otherwise, please go and check that session obviously after the class. Okay, because here in the tissue system, right, in the under the topic tissue system, I have covered maximum things. Even the exceptions there. Okay, so that will be very good for you to cover that particular part. Okay, so now first of all, how are we going to start today? Today we are going to start with the dicot stem first. Clear? We are going to start with the dicot stem first. So are you guys ready for this? Yes. What are we going to start with? We are going to start with the internal structure of the dicot stem first, anatomy of the dicot stem first. Now just look at this diagram. Okay, or look at this diagram. It is directly from NCERT. So what you people can say? What you people can say? Now already in the tissue system we have covered it, but still just look at this diagram. Look at this diagram and tell me what you people are going to write about the dicot stem. And how can you identify by looking at the section that it is the dicot stem? Whatever points are there in your mind, just let me know. Whatever you know about the dicot stem or about the stem section, tell me in the chat section, students. And please, there should be the interaction. So all of you, it's a request, guys. Please start reporting in the chat section. Quick. Yes. Quick. Double what? Give it a try. Quick. Try to answer this question. It is important as well. You know it very well, right? So when it comes to the anatomy of the dicot stem, okay. So first of all, if it is a stem, the vascular bundles, what type of vascular bundles will be there, right? Conjoint, collateral, and it is a dicot stem. So obviously open vascular bundles will be there, isn't it? It's conjoint, collateral and open vascular bundle. So what is the meaning of conjoint, collateral and open vascular bundle? That here, bache, you will see the cambium in between, right? We will be having the xylem and the phloem, right? Xylem and the phloem, and in between xylem and phloem, there will be the cambium. What will be there, bache? There will be the cambium. Are you getting my point? Are you getting my point? There will be the cambium. Right, so these are the conjoint, collateral, open vascular bundle. So what is the meaning of this conjoint, collateral? Your xylem and phloem, it is present on the same radius. Okay, xylem and phloem, it is present on the same radius. And when it is the open vascular bundle, means it is open for secondary growth, right? The secondary growth is possible there. And you know that for secondary growth, this cambium is required. And for this cambium, we also use the word intrafascicular cambium. Intra means within, of course, and it is also known as fascicular cambium, isn't it? Fascicular cambium, right? So this is the first point that you should know when you know that you should know about the stem part. And secondly, bache, the arrangement of xylem will be in dark here. What type of xylem will be there? The xylem arrangement will be in dark. Yes or no? The xylem arrangement will be in dark. Quick. Quick. Done? Yes, so it is intrafascicular cambium, also known as fascicular cambium, and here you people can see, right, that this type of vascular bundles are there. Now when you look at the section of the stem, you can see that these vascular bundles, they are arranged in the form of a ring, bacho. How are they arranged? They are arranged in the form of a ring. Just look at this diagram and explain these points, right? Here the pith is large, it's conspicuous. Pith is large, it's conspicuous. Okay, and when you will see the cortex here, the ground tissue here rather, it is having hypodermics, hypodermis, general cortex, endodermis, pericycle, all of them are present there, right? So this is about the stem. So first of all, when it is a stem, the vascular bundle will be conjoint collateral, and if it is a dicot stem, it is going to be open type of vascular bundle. So now let's talk about the dicot stem. So how are we going to start first? Obviously we are going to start with the epidermis first, isn't it? We are going to start with the epidermis first. So students, when it is epidermis, outermost layer, now it is a stem. So tell me, is it covered with the cuticle or not? Quickly, is it covered with the cuticle or not? We are talking about the epidermis here, right? So epidermis is no doubt outermost layer, yes or no? It is outermost layer, yes or no? Tell me quickly, what is this epidermis? It is the outermost protective layer. Right, it is the outermost protective layer, and obviously it is covered with cuticle, yes or no? It is covered with cuticle, yes or no? Tell me quickly. We know that we have the outermost. Just allow me a minute, students. Just allow me a minute. Okay. So, yes. So what is it? What is it? It is the outermost protective layer which is covered with what? Which is covered with the cuticle. Right, bache? And here, because it is the epidermis, you know that it will contain stomata as well. Yes or no? And it will be having the epidermal appendages. Okay, it will be having the epidermal appendages. So because it is, yes, it will be having what? It will be having the epidermal appendages. So because bache, it is what? It is the epidermis of the stem. So you know that here you will see what? You will see trichomes. What will be there? There will be the trichomes. So trichomes, right? It is multicellular, and what is the function of trichome? To reduce the water loss. Yes or no? To reduce the water loss. Yes or no? And cells, of course, they are single layered and they are compactly arranged. There will be no intercellular spaces here. Clear, bache? Yes or no? So stomata will be there, trichomes will be there, cuticle is covering the epidermis, right? Trichome will prevent the water loss, right? So this is about the epidermis, isn't it? This is about the epidermis. Now next is what? Next is the cortex. So because it is a stem and it is a dicot stem, so cortex is divided or not? Like cortex is multi-layered or not? Or cortex is divided into different layers or not? Tell me quickly. Let him spam. It's okay. It is such students, they are basically not the students. They are just here for the spamming. So ignore them, right? Let him do whatever he is doing. It should not affect us, right? So you quickly tell me. Cortex. What about the cortex here? What about the cortex here? Will it be divided or not? Will it be divided or not? Yes. So seriously, sunlight, I'm talking about the dicot stem, and you are saying in dicot stem spongy and pellucid parenchyma is there. No, that is the part of a leaf. So Deena, it is divided into, yes, it is divided into, and Deena, ask others to join the class as well. Yes, so here the cortex is divided into your hypodermis. Okay, it is divided into hypodermis. It is divided into general cortex, and here you will also see endodermis, and then bache, then in the case of dicot stem, pericycle and pith is also there. Okay, so if it is a dicot stem Samaria, here what will you see? The hypodermis will be there. So hypodermis means the layers just below the epidermis. Okay, the layer just below the epidermis is your hypodermis. Clear, bache? The layer just below the epidermis is your hypodermis, and you know that it is having, right, this hypodermis is multi-layered. Fine, it is multi-layered, and because it is a dicot stem, so it is having collenchyma. Fine, it is having collenchyma. Clear, bache? It is having collenchyma. Is it clear? Yes or no? Is it clear? Quickly tell me. Is it clear? Quick. Done? Sure. So hypodermis is multi-layered here, having collenchymatous cells. Clear, bache? Having collenchymatous cells, and these collenchymatous cells are also having the chloroplast, so it is also helping in the photosynthesis. Clear, bache? So just look at this diagram. So collenchymatous hypodermis is there in the case of dicot stem. Done, bache? And here, when you look at this part, this is the general cortex. Okay, what is it? It is the general cortex. Now, bache, when you see this general cortex, so you know that in the cortex, mainly we have the parenchymatous cells. What type of cells do we have? Mainly, we have the parenchymatous cells. And you know that what is the function of parenchymatous cells? The main function of these parenchymatous cells is to store the food, right? And here, even in these parenchymatous, in this cortex, right, some of the parenchymatous cells, they are also having the chloroplast. So, remember it, right? Please focus here. Below epidermis, you have the hypodermis that is made up of collenchyma. That is made up of collenchyma. Now, below this hypodermis, this part is the general cortex here. Yes, this part is the general cortex here. And here in this general cortex, some cells, some parenchymatous cells are also containing chloroplast, and that is why you are calling them as chlorenchyma. So do not confuse these two words. Okay, do not confuse these two words. Is that clear? Yes, bache. Is that clear? Yes or no? Yes or no? Tell me quickly. Yes or no? So this is what you need to write there in the general cortex, right? This is what you need to write there in the general cortex. That general cortex is also multi-layered. It is also multi-layered, having parenchymatous cells. Okay, having parenchymatous cells, and some cells are having chloroplast. So obviously they are performing the photosynthesis, and others are storing the food. Okay, so they are even storing some raisins, tannins like this as well. Fine. So now the next to it is what? It is the endodermis, and you know that when it comes to the endodermis, it is the inner layer of cortex. What is endodermis, students? It is the inner layer of cortex. What is it? It is the inner layer of cortex. Because it is a dicot stem, so here endodermal cells are having starch. Okay, and that's why, so we call it as starch sheath. Done? This is what you need to remember, because it is a stem. So here endodermis is not having the Casparian strips. Endodermis is not preventing the water movement, right? Because Casparian strips are not there. So in the case of dicot stem, your endodermal cells, your endodermis is known as the starch sheath. What is it, bache? It is the starch sheath. Why? Because endodermal cells are having the starch grains. Okay, they are fully loaded with the starch grain. That is why. Is it clear, bache? Is it clear? So here you guys can see the endodermis, the epidermis, hypodermis, and this one is your general cortex. And you can see here, bache, the cells are having the intercellular space. Okay, intercellular space is also there in the cells. Fine, bache, intercellular spaces are also present there. Any doubt? Clear? Any doubt? Sure. So next to endodermis will be, yes bache, yes Deena, next to endodermis will be pericycle. So you know that pericycle, vascular bundle and pith, they are making the steel. Okay. Pericycle, vascular bundle and pith, they are making the steel. What are they doing? They are making the steel. So next to endodermis is what? Next to endodermis is the pericycle. Okay, it is the pericycle. And please note down, students. Fine, please note down. So now just look at this diagram. See that this one is the star sheath here. So now when you look at the pericycle, can you see that pericycle here? Right? Can you just see that? This pericycle here? Yes or no? Just look at the diagram. Look at the diagram here. So pericycle, of course, you know that pericycle is present below the endodermis, right? Pericycle is present below the endodermis, and here if you look at the pericycle, so it is made up of what type of cells, bache? Can you see that? See, it is made up of sclerenchymatous cells. Okay, it is made up of sclerenchymatous cells as well. Like in some plants, you will see that this pericycle, it is bache multicellular. What is it? It is it is multi-layered, right? It is multi-layered. And if you will look at this patch, can you see this patch here? Can you see this patch here? It is the sclerenchymatous patch. What type of patch it is? What type of patch it is? Right, it is the sclerenchymatous patch. And sometimes in between in this pericycle, you will see sclerenchymatous patches, then parenchymatous patches, sclerenchymatous patches, parenchymatous patches. So this is how, right? This is how this pericycle is present there. Clear, bache? Clear, bache? So you will see the alternate bands here. Fine. You will see the alternate bands here. Yes or no? Yes or no? Yes or no? So now how to remember this? Wait. So endodermis, epidermis, hypodermis, general cortex, then comes your endodermis. Okay. So next to endodermis is your pericycle. That's what I'm saying. So bache, when it comes to this pericycle, so next to it is your vascular bundle. And now because it is a dicot stem, so dicot stem mein what type of vascular bundle will be there? It is a dicot stem, so what type of vascular bundle will be there? Quickly type it in the chat section. What type of vascular bundle will be there? Because it is the, yes, it is the dicot stem. So obviously here the vascular bundle is open type. है ना? It is, the vascular bundle is open type. So here you guys can see the cambium, your intravesicular cambium, the phloem and the xylem here. So xylem is endark because protoxylem is facing the center, it is facing the pith. Now this pericycle, just suppose this here you have the endodermal cells. Right, here you have the endodermal cells. So just below endodermis, here we have the pericycle. Right, here what do we have? Here we have the pericycle. So when you look at the diagram, even from your NCERT, you will see these kind of patches. So when we draw cells like this, so we are trying to say that these are the sclerenchymatous cells. Okay, when we draw it like this, so we are trying to say that these cells are sclerenchymatous. And you know that sclerenchymatous means they are dead, they are lignified, they have thick cell wall, isn't it? It means that they are dead. Okay, they are lignified, they have thick cell walls. So sclerenchymatous patches are there. So these sclerenchymatous patches are present just opposite to this phloem. Is it clear? Right. So if I talk about the NCERT, in the NCERT it is clearly written that semi-lunar patches, sclerenchymatous patches are present in pericycle. Yes or no? Semi-lunar, half moon, like this, half moon, okay, like this. So that is how your pericycle is present. Fine. So sclerenchymatous patches are there. Sclerenchymatous patches are there. So bache, sometimes you will only see sclerenchymatous patches, but here in between these sclerenchymatous patches in the pericycle, you also see the parenchymatous cells. Okay, you also see the parenchymatous cells. right? Right, so sclerenchymatous patches, sclerenchymatous patches, sclerenchymatous patches. In between, these are the parenchyma cells. So like in sunflower, okay, you can take the example of sunflower. So in sunflower, you will see that this pericycle is heterogeneous. What is the meaning of being heterogeneous? Right, it is having sclerenchyma as well as parenchyma, the way I'm explaining. Okay, so it is having sclerenchyma as well as parenchyma. Fine, it is having sclerenchyma as well as parenchyma. Is it clear? Is it clear? So this is what you need to keep in your mind. Done, bache? This is the pericycle. This is the pericycle. So just next to this phloem, you will find these sclerenchymatous cells of the pericycle. So just look at it. Clear, bache? Just look at it. Sclerenchymatous patches, then parenchyma. Sclerenchymatous patches, then parenchyma. And here, how these vascular bundles are arranged? In the form of a ring. Okay, in the form of a ring they are arranged. Clear, bache? Clear, bache? That's the point. Done? That's the point. So obviously, again phloem, cambium and the xylem will be there. And the pith is large. It's conspicuous. Okay, pith is large, it is conspicuous. And bache, here in between this pith, this part here, in between this vascular bundle, this part is the medullary ray. Okay, it is also known as the vascular ray. It is the medullary ray or the vascular ray. So it helps in the radial conduction of the water. Clear, bache? Radial conduction of the water. Done? Done? So that's how it is present. So any doubt here in the dicot stem? Tell me, any doubt here in the dicot stem? Then we will revise it from NCERT, of course. Okay. We will revise it from NCERT, of course. Sure? Done? Okay. Chaliye. So let's revise it from, that's all, bache. That's all. You can read it from NCERT by your own. Why to waste time here? So this is about the dicot stem. So look at the diagram and explain the topic. Look at the diagram and explain the topic. That's all. So if there is any doubt, do let me know. And yes, in your notes, you can include one more thing, that when you talk about the cucurbita, okay, when you talk about the members of Cucurbitaceae family, okay, in Cucurbitaceae family, when we talk about the stem is having 10 vascular bundles. No doubt they are arranged in the form of a ring. 10 vascular bundles are there. They are arranged in the form of a ring, and these vascular bundles are conjoined. They are conjoined, means they are joined together, but they are bicollateral. Okay, they are bicollateral. So they are conjoined, bicollateral and open. In Cucurbitaceae family, what type of vascular bundles are there? No doubt they are conjoined. Xylem and phloem is present on the same radius. They are joined together, right? And it is bicollateral. Xylem is there in the center. Phloem is, right? Phloem is covering it from both the sides. Clear, bache? And it is open, means secondary growth is possible. This is what you need to keep in your mind. And yes, one student was asking this question. Yes, you are right. The medullary ray here, it is made up of parenchymatous cells. It's a fact. Right, it is made up of parenchymatous cells. So next is, bache, anatomy of the monocot stem. Again, here also you will see epidermis. Right, here also you will see epidermis, and again it is also the outermost protective layer covered with the cuticle. Right, outermost protective layer covered with the, fine, covered with the cuticle. Yes or no? Outermost protective layer covered with the cuticle. Here the story is again same. Fine, right? But when you look at this part, right, can you see that cortex is not, right, that cortex is not properly differentiated? When you look at the monocot stem, see, have a look here. This is the section, this is the transverse section here. So can you see that vascular bundles are scattered here? Yes, vascular bundles are scattered. They are just scattered. Yes or no? Vascular bundles are just scattered. And if you look at this particular diagram, see, periphery. Periphery is having small vascular bundle. Right? Vascular bundle near to the periphery, they are small but more in number. Right? They are small but more in number. And see the central part. here vascular bundles, they are large but few in number, right? They are large but few in number. That's what you need to keep in your mind. Okay, and that's what we have already covered in the last class. If you have any doubt, do check it out. Clear, bache? Do check it out. And moreover, when you look at the monocot stem, bache, because it is a monocot stem, you know that there is no secondary growth. Okay, there is no secondary growth. So your vascular bundles, no doubt, they will be conjoined, they will be collateral, but closed type of vascular bundle will be there, right? So there will be no cambium there. There will be no cambium there. So conjoined, collateral but closed. Conjoined, collateral but closed. And you can see here the cortex is not differentiated into general cortex, endodermis, pericycle. So that general cortex, endodermis, pericycle, they are absent in the case of monocot stem. Clear, bache? They are absent in the case of monocot stem. Clear? Clear, bache? So that is what you need to keep in your mind. But yes, hypodermis is there. So below epidermis, there will be the hypodermis. Below epidermis, there will be the hypodermis, and it will, and this hypodermis is multi-layered, of course. It is multi-layered, but it is made up of sclerenchyma. So it is multi-layered and sclerenchymatous. Okay, it is multi-layered and sclerenchymatous. Are you getting my point? It is multi-layered and sclerenchymatous. Clear, bache? Clear, bache? Okay, so obviously it will provide the mechanical support. It is going to provide the mechanical support. Clear? Done. So here when you talk about the ground tissue, no doubt ground tissue is large, but ground tissue is not differentiated. Ground tissue is not differentiated into general cortex, endodermis, pericycle, or medullary rays, right? So ground tissue is not differentiated like this. Fine, bache? Ground tissue is not differentiated like this. Fine. So ultimately, no doubt, ground tissue is there. It is large. It is parenchymatous. Okay, it is large. It is parenchymatous. It is conspicuous, but it is not differentiated there. Okay, bache? It is not differentiated there. Done, bache? Yes? Okay. Done. So vascular bundles are scattered. I told you already, right? So center mein they are large in size, less in number. Periphery pe small in size, more in number. So conjoined, collateral, closed type, and xylem again is endark type. Clear, bache? Xylem again here is what type? Xylem again here is the endark type. Okay? Done. So now, if you observe here, see this protoxylem. This part is the xylem, na? This part is the phloem here. So xylem mein, see this protoxylem is facing the center. So obviously the arrangement here is endark. But bache, if you observe this, if you observe these vascular bundles, let me show you this part. Okay. This is the vascular bundle. So can you see that this vascular bundle is also covered with a kind of sheath? Yes, it is covered with a kind of sheath. So here it is covered with this sclerenchymatous bundle sheath. Okay, it is here covered with the sclerenchymatous bundle sheath. So bundle sheath is nothing. It is a layer, right? It is a layer of cell which is covering the vascular bundle. What is this bundle sheath? It is the layer of cells, right, which is surrounding the vascular bundle, and here it is sclerenchymatous. This is what you need to keep in your mind. That's all. Okay, that's all. Raju bache, how, what are you getting for doing it? What are you getting? From last 38 minutes, you are wasting your time. So are you that free? Huh? Are you that free? That I'm doing the spamming here, see emojis, and now see I'm typing this. What are you getting? इतने फ्री हो क्या? कोई और काम नहीं है क्या? मतलब बिना बात के कुछ नहीं है संडे को, क्या करें? चलो, let's go and start spamming. Useless you are, right? Literally useless. Done, bache? So this is about the monocot stem. Okay, This is about the monocot stem. So here you people can see the comparison as well. Dicot stem, monocot stem, hypodermis, collenchymatous, sclerenchymatous. Ground tissue differentiated, not differentiated. Starch sheath means your endodermis present, absent. Medullary rays present, absent. Vascular bundle, collateral open, collateral closed. Arranged in a ring, scattered. Okay, secondary growth occurs, secondary growth doesn't occur. That's all. So these tables are the most important thing here. Right, bache? Okay, clear? So next is the dicot root and the monocot root. Okay, next is what? Next is the dicot root and the monocot root. Bache, in dicot root, okay, secondary growth is there. Okay, here secondary growth occurs, but that doesn't mean that you will have open type of vascular bundle here, because it is the root. So when you talk about the roots में what type of vascular bundle is there, बच्चे? R for roots, R for radial. R for root, R for radial. And what type of xylem arrangement will be there? That is exarch. Okay, that is exarch. What is it? That is exarch. Clear? Clear? So here you guys can see that your protoxylem is facing the periphery. Okay, and see the xylem is present. Here the phloem is present here, so obviously they are present at different radii. They are present at different radii, and what type of arrangement is there? Exarch arrangement is there. And can you see the hypodermis here? Yes. Can you see the hypodermis here? Tell me. Can you see the hypodermis here? Yes or no? Of course not. So whether it is a dicot root or a monocot root, in the roots hypodermis it is absent. In the roots hypodermis is absent. Clear, bache? So first of all let's start with the epidermis. But here instead of epidermis we used to call it epiblemma. Okay, we used to call it epiblemma or even the piliferous layer. Okay, piliferous layer. Piliferous layer or piliferous layer. So epidermis, right? So obviously it is the outermost layer, but here not covered with cuticle. So in the roots epidermal cells are not covered with cuticle. Clear, bache? They are not covered with cuticle. And moreover stomata, because from roots there will be no loss of water in the form of gases, no transpiration is there. Right, so stomata is absent. Stomata is not the part of this epidermal tissue system in the case of roots. Okay, stomata is absent. And when you talk about the epidermal appendages here in the case of roots, yes, bacho, when you talk about the epidermal appendages here in the case of roots, so they are root hairs. Okay, what are they? They are root hairs. And what are they? They are unicellular. And you know that what is the role of root hair? They are meant for the absorption of water and minerals. Okay, they are meant for the absorption of water and minerals. So it is the epiblemma here. What is it? It is the epiblemma here. Clear, bache? Clear, bache? So I am writing it again. So hypodermis is absent. Hypodermis is absent in roots. Clear? Hypodermis is absent in roots. Done, bache? Sure. So next to it is cortex. Fine. So cortex may, you know that parenchymatous cells are present. What is there in the cortex? Parenchymatous cells are there or not? Yes or no? So it is the cortex. Cortex is made up of thin-walled parenchymatous cells. Thin-walled parenchymatous cells having intercellular spaces in between. Having intercellular spaces in between. Are you getting it? This is what the cortex is. Yes or no? So obviously roots are not going to do the photosynthesis. Okay, roots are not going to perform the photosynthesis here. So of course, when you talk about the cortex, right, these parenchyma cells, they do not have the chloroplast. Okay, they do not have chloroplast, because in the case of roots, we will not see the photosynthesis. Okay, in the case of roots, we are not going to see the photosynthesis. But yes, again, bache, we have the exceptions as well. Do you know that we have some photosynthetic roots also? Even in the morphology, this example is given. Photosynthetic roots are also there. That is your Ctenospora and Trepa. Right, Ctenospora and Trepa. They are the examples where roots are photosynthetic. So if roots are photosynthetic, then obviously the cortex will contain the chloroplast. Okay, cortex will contain the chloroplast. Is it clear? Yes or no? Is it clear? Yes or no? So next to it is what? Next to it is your endodermis. Yes or no? Next to it is your endodermis. So you know that here endodermis is playing a major role. So endodermis, remember this point always. It is the innermost layer of the cortex. What is it? It is the innermost layer of cortex. Fine. It is the innermost layer of cortex, endodermis. So now here in the endodermis, what will we see? Endodermis have Casparian strips. What type of strips are there? Casparian strips are there. Casparian strips are there. And these strips are water impermeable. Okay, what are they? They are water impermeable. Why? Because they are suberized. What is the meaning of being suberized? There is the deposition of the suberin. Okay. Suberin is deposited there. There is a deposition of the suberin. So suberin is water impermeable. It will not allow the water to move from that endodermal cells, right? So this part we will discuss in the transport in plant chapter, right? In the plant physiology, where you will see that plants, they form two pathways for the water movement. One is the apoplast, which is from the dead spaces, from the intercellular spaces, from the secondary cell wall. Another is the symplast, which is there from the cytoplasm. So because here in the endodermis, Casparian strips are present, so they are forming a kind of, they are forming a system where water will not be, where water cannot cover these endodermal cells because of these Casparian strips. So it needs to, so at this point, what is going to happen? The water will take the symplastic pathway. So this apoplast and symplast we will discuss in the plant physiology. Clear? We are going to discuss that in the plant physiology. So Casparian strip, water impermeable, suberized, suberin is deposited. Clear, bache? That is why it is suberized, right? And where are these strips present? On the radial and the tangential wall of the endodermis, isn't it? On the radial and on the tangential wall of the, yes, they are present on radial and tangential walls of the endodermis. Okay. Chalo, let me try. This is suppose right here the tangential walls and the radial walls will contain the, right, radial walls and tangential wall will contain the, yes, Casparian strip. So this is what you need to keep in your mind. So it will act like a water impermeable system, right? Why? Because suberin is deposited there. Okay, suberin is deposited there. Now, bache, you know this arrangement, isn't it? You know this arrangement, isn't it? That we have the endodermal cells here. Let's focus on this vascular bundle wala part first. So we have the endodermal cells here, then the pericycle is there. Okay, and then you have the xylem and phloem. So that is how xylem is present, the protoxylem, and then comes the metaxylem. And here you will see the phloem. The cell shape is of course not proper, but we'll tell you. Okay. That's how it is present. So it's a dicot root. What is it? It's a dicot root. See, bache, you can even compare a dicot root with the dicot stem. So if you will see the dicot root, the pith is not so large, but in dicot stem it is so large, right? The pith is not so large when you talk about the dicot stem, isn't it? Isn't it? The pith is not so large when you talk about the, yes, when you talk about the dicot root. And pith is very large, it is conspicuous in the case of dicot stem, you know it, right? So now you know that because it is a root, the xylem is arranged. What type of arrangement will be there? It is exarch. What type of arrangement will be there? It is exarch. So you guys can see here, right? You people can see here the protoxylem is facing the periphery. So bache, this protoxylem, now see, this is the endodermal cell that is faced by the protoxylem, right? Protoxylem is facing this particular endodermal cell. So the cell which is right near to this protoxylem, okay, the cell of endodermis right, the cell of endodermis which is just near to this protoxylem, right, it is the passage cells, bache, it is a passage cell. It doesn't contain the Casparian strips. Clear? Clear. So you can say that the cell of endodermis which is situated in front of protoxylem does not contain Casparian strips, so known as, right, Casparian strips, so known as passage cell. Clear? It is known as passage cell. That's what you need to keep in your mind, right? And when it is the dicot root, obviously if you will see the xylem and phloem, total number of vascular bundle here, so you can see it is right from 2 to 6, right? 2 to 6 vascular bundles can be there. So diarch to hexarch, like mostly it is tetrarch, but here when it comes to the arrangement of these vascular bundles, especially the xylem, 2 to 6 bundles are present. So it is diarch to hexarch when it is the dicot root. Clear, bache? When it is the dicot root. Is it clear? Yes or no? Yes or no? Tell me. It is diarch to hexarch. Done, bache? And the pericycle, it is again parenchymatous. Fine. Pericycle is what? It is again parenchymatous. Done, bache? That's all. So few cell layered thick, parenchymatous it is. Now yes, one more point. Bache, you know in the case of roots, we also draw the lateral roots also. We also draw the lateral roots also. In the case of roots, we also draw the lateral roots. So lateral roots arise from pericycle. Do you know that lateral roots arise from pericycle? That's why it is endogenous in origin. Because lateral roots, they arise from pericycle. That's why we used to say they are endogenous in origin. What are they? They are endogenous in origin. Clear? They are endogenous in origin. Done? Any doubt? Yes. Any doubt? So pericycle, parenchymatous, few cell layered thick, and it will form the lateral roots. That is why in the case of roots, we always used to say that lateral branches are endogenous in origin. But if you talk about the stem, there the lateral branches are exogenous in origin, means they are not arising from the pericycle. Okay, they are not arising from the pericycle. That's the point here. Done, bache? That is the point here. Okay, so pith is small, less developed, it is inconspicuous or absent. Okay, so vascular bundle, you know that they are going to be radial and exarch, usually 2 to 4. Okay, usually 2 to 4, but it can be 6 as well. That's why I'm using the word diarch to the hexarch. Usually, but they are 2 to 4. Fine. But up to 6 you guys can see in the case of dicot root. So see, the section is so easy. है ना? This section is so easy. Now compare this with the, okay, see, this is the diagram given in NCERT. Fine. So compare it with the monocot roots. Compare it with the monocot roots. So obviously major differences of vascular bundle. Here you will see many vascular bundle and you will see the pith is large. So here I can say that it's polyarch. So many vascular bundles are present here. Yes, it is polyarch because so many vascular bundles are present here. So if you compare dicot root with the monocot root, so you will see that pith is large and conspicuous. But in the dicot root, right? But in the dicot root, it will be small. And when it comes to the arrangement of the vascular bundle, it is polyarch. Rest everything is same, bache. So here you guys can see the comparison. It is actually the root, not the stem. So right, so dicot root may, and yes, how can I forget? So now see this part in between. This is the phloem, this is the xylem. So this part in between, this is parenchymatous. Okay, this part here is parenchymatous. Actually it is known as the conjunctive tissue. What is it? It is known as the conjunctive tissue. And when there will be the secondary growth in the case of dicot root, this conjunctive tissue will become meristematic. It will start forming the cells, right, bache? It will help in the formation of the cambium then. Clear? It will help in the formation of the cambium then. Clear, bache? Clear, bache? So that's all. So cambium, so conjunctive tissue, parenchymatous it is, cells are differentiated into vascular cambium. Because in monocot root there is no secondary growth. So here you will see that this conjunctive is mostly sclerenchymatous, sometimes parenchymatous, and it will not form vascular cambium. Here when you talk about the cambium, it is the secondary in origin. And here cambium is absent. So I will share the notes in my Telegram group, that is Love Biology by Ambika. You guys can join that Telegram group and you will get the PDF notes there. So this is all about the dicot root and the dicot stem. And here you guys can see the comparison in between monocot root, dicot root, monocot stem, dicot stem as well. So as I said, I will share it in the Telegram group. So next is bache leaf. Right, the anatomy of the leaf. So in the case of leaf, you know that leaf is the major organ for the photosynthesis, right? So cells are fully loaded with the chloroplast here. Okay, it is a major site of photosynthesis. And when you talk about the leaf, here the ground tissue system, here the ground tissue is known as the mesophyll basically. What is it? It is the mesophyll. What is it? It is the mesophyll. And even in the case of leaf, you will not see secondary growth. So here the vascular bundles will be conjoined, collateral, and closed type of vascular bundle will be there. Okay, closed type of vascular bundle will be there, right? Right. So that is what we need to see here in the case of the leaf. Okay, bache? Done? Yes or no? Yes or no? Any doubt here? Yes? So here when you look at the epidermis, see, we have two type of leaves. One is your dicot, another is your monocot. One is your dicot, another is your monocot. D for dicot, D for dorsiventral. Here the leaf is dorsiventral, right? Both the faces are not same, they are different. The stomata distribution, the type of cells they are present, they are different here, right? And now in the case of monocots, the leaves are isobilateral, right? Or even they are also known as equifacial. Both the sides are same. And even if I compare it with the venation, I hope you people remember, in the case of dicots, mostly you will see reticulate venation. Bache, do you remember this part? Mostly you will see reticulate venation, network of veins, and veinlets will be there. Okay, so accordingly vascular bundles are distributed. And when it is the monocot, in the case of monocot, you will see the parallel venation. What will you see there? You will see the parallel venation there. That is how the vascular bundles are distributed. Okay, so in the case of dicot, as I said, dorsiventral leaves are there, right? Dorsiventral leaves are there. Both the sides are different, bache, and here both the sides are same. So now when you look at the section of the dicot leaf, here you will see, right, both the surfaces are having the epidermis. Clear? Both the surfaces are having the epidermis. What do they have? They have the epidermis, both the sides, and this epidermis is even covered with the cuticle. Why is it so, Dina? Why is it so? Why epidermis of both the sides it is covered with cuticle? So that there is less water loss. Okay, so that there is less water loss. And you know that here you will see stomata as well. What are you going to see here? You are going to see the stomata there. Okay, stomata there. So when it is the dicot, in the case of dicot, upper surface is also known as ventral surface or the adaxial surface. Let me tell you. Upper surface is known as the ventral surface or the adaxial surface. This is the upper surface. Here when you look at the mesophyll, see, mesophyll mein two type of cells are there. You can see the elongated cells also. See, elongated cells. This is the palisade parenchyma. What type of parenchyma it is? Palisade. Elongated cells having the chloroplast. Elongated cells having the chloroplast. And when you look at this lower side, see, can you see this type of parenchyma? It is spongy parenchyma. And here in between you can see the air spaces also. Both the sides are having stomata, but in dicot leaves, the mesophyll cells, their arrangement is different. The upper surface is also known as the ventral surface. It is also known as the adaxial surface. Adaxial it is, not abaxial, it is adaxial. Adaxial. And this one is the lower epidermis, means dorsal surface, means abaxial it is. It is abaxial surface. Right? What is it? It is the abaxial surface. So spongy parenchyma is towards the abaxial or the dorsal surface or the lower surface. Clear? Or the lower surface. Done? So both of them contains the chloroplast. So chloroplast is there, means the photosynthesis will occur. Okay, the photosynthesis will occur. Is that clear? Yes? Is that clear? Sure. So when you look at the vascular bundle, I told you already, conjoint, collateral and closed. What type of vascular bundles are there? Conjoint, collateral and closed. Right? Conjoint, collateral and closed. Yes or no? Repeated, bache. Conjoint, collateral and closed. Vascular bundles are same. Okay. Even in your monocots, even in your dicots, when you look at the vascular bundle, they are going to be same. Conjoint, collateral, closed. And bache, protoxylem. Protoxylem faces upper epidermis. That is your upper epidermis. Right? It faces what? It faces the upper epidermis. And obviously your protofloem, the phloem which forms first, it faces the lower epidermis. Done? It faces the lower epidermis. Clear, bache? Protoxylem situated towards the upper epidermis, that is adaxial surface, and protofloem towards the lower epidermis, that is abaxial surface. Clear, bache? So no endodermis and pericycle is there. No endodermis and pericycle is there. And yes, here also you will see the bundle sheath. Bundle sheath means the covering of these vascular bundle. So bundle sheath will be parenchymatous. Okay, bundle sheath will be parenchymatous. That's all. So this is about the dicot leaf, where there is the palisade and the spongy parenchyma is there. Where there is the palisade and the spongy parenchyma is there. Clear, bache? Now come to the diagram from NCERT. I told you already about it, right? So next is the monocot leaf, isobilateral. From both the sides it's all same, right? And yes, in the dicots na, even the stomata distribution, it is different. Just lower side will have more stomata, upper epidermis will have less stomata. Why lower side is having more stomata? So that water loss due to transpiration will be less. Gaseous exchange will be there proper, but the water loss due to the transpiration will be less. So the lower side is having more stomata, and upper side sometimes stomata are absent there. If they are present, they are very less in number. Clear, bache? But in your isobilateral leaves, both the sides are having same stomata. So what you need to remember here in the case of monocots, that their epidermis contain motor cells or the bulliform cells. What type of cells are there in the epidermis of the monocots, right? Especially your grasses, you will see the motor cells or the bulliform cells. Now, what type of cells they are? See, bache, when water is present in sufficient quantity, then these cells will become turgid. Okay. When the water is present in the sufficient quantity, these cells will be turgid. They are turgid, the leaf surface will be exposed. But when there is the water scarcity, these bulliform cells, they will lose the water, right? They will become flaccid, or you can say that shrinked. So the leaf, there will be the curling of the leaf surface. Okay, what will be there? There will be the curling of the leaf surface to protect, and even that curling is also helpful. Na? The water loss due to transpiration will also be less. Clear? And here in the case of monocots, you are not going to see the palisade and spongy parenchyma. It's all same. Fine, it's all same. And see next to these bundle sheath cells, you will see some patches of these sclerenchymatous cells. That's all. Fine, that's all. Done? Any doubt? Bundle sheath is parenchymatous, but it is further covered with the sclerenchyma. Done? So intercellular spaces are less. So these distinct patches of sclerenchyma are present above and below the vascular bundle. That's all. Okay, that's all. So vascular bundle sheath is parenchymatous. Always remember, bundle sheath will be parenchymatous. It is made up of parenchyma only. But here you will see some extensions. Here you will see some extensions of bundle sheath which are sclerenchymatous, and you guys are going to see it in the case of monocot leaf. That's all. And yes, mesophyll is not differentiated here. So this is the diagram from NCERT. Clear, bache? Clear? So here again you guys can see the comparison of a monocot and the dicot leaf as well. All the points are given here, and right, and these are the diagrams from the NCERT. So I hope all your doubts are clear. Done, bache? So just look at this diagram, how in the form of rings these vascular bundles are arranged, right? So epidermis, collenchymatous hypodermis, then parenchyma, endodermis, pericycle. So here it is not shown properly, but you know that pericycle will be sclerenchymatous, right? So it can have mixed sclerenchyma and parenchyma as well, but these semilunar patches are present. Then phloem, then cambium, and protoxylem here is facing the pith. So in dicots, you can see that dicot stem, the pith is large and conspicuous. The pith is large and conspicuous, and monocots, it's scattered, right? Hypodermis is sclerenchymatous, and these are the diagram of the leaf. Clear? So next is the secondary growth in the dicot stem, and in these 3 steps we have to discuss this particular topic. Okay? Any doubt? Now the doubt time. Ananya, Subhashini, Agansha, Deena, Ishwat, any doubt from these sections? I told you the section part is very easy. Okay. And I told you this thing before, the section part is very easy. If you know the tissue system, then it is very easy. So anatomy, please do not leave the anatomy for your NEET paper. Okay. It's very scoring. Anatomy is actually very scoring. So now we have to start the secondary growth. So do you find this topic easy or difficult? Yes. Do you find this topic easy or difficult? But there is no need for elaboration, Ishwat. All you can remember is in the epidermis of monocot, especially in the grasses, you will find these special cells, some modified epidermal cells they are, right? So they will obviously, it's like a coping mechanism for the grasses. If there is a water loss, obviously that cells will lose the water, they will fold the leaf surface. So that water, right? So that water loss will be less. Clear, bache? Clear? It's okay, Deena. I was expecting the same from you, from your batch. It's fine. Chalo. So secondary growth in dicot stem, this is what we need to talk about, and we will discuss it in 3 steps, right? We will start with the formation of cambial ring, activity of cambial ring, then activity of cambium, and Yes, formation of cork cambium and then the activity of cork cambium. Done? So formation of cambial ring, activity of cambial ring, then formation of cork cambium and activity of cork cambium. That's how we need to discuss it. So if there is any doubt, please tell me. Quick, if there is any doubt, please tell me. Done, बच्चे? Okay, so do you find this topic difficult or easy? And yes, let me tell you, bache, because of some security reasons in Jammu and Kashmir, the internet is not working. So your Wazeem sir will not be able to take the class today and tomorrow. Today and day after, I think tomorrow and day after tomorrow, he won't be able to take any class. Fine. Done. Chalo. So now let's start this particular topic, that is formation of cambial ring, activity and the activity of the cork cambium. So what do you know about the secondary growth? Here, bache, no doubt we need to follow the NCERT only. This is a diagram that we need to understand actually. So what do you know about this topic? Or do you find it difficult or not? Or for you it is very easy or something? Tell me. Tell me quickly. Do you know anything about this topic or you don't know? What do you know about this topic? Tell me. I'll make it very easy for you. And yes, this topic is easy. It is very easy. Chalo, let me explain this. We will start with the vascular bundles. How are we going to start this? We are going to start it with the vascular bundles. Clear, bache? We are going to start it with the vascular bundles. Okay, so you know that when it comes to the secondary growth, so for the secondary growth, what do we need? We need the secondary meristem first. Yes, we need the secondary meristem first. Yes or no? There should be the formation of the secondary meristem first. Now, what is the secondary meristem? Okay, we have discussed it before, bache. So, you know that the primary meristem is the one which will form during the embryonic growth, isn't it? Primary meristem is the one which will form during the primary growth of the plant. It helps in the elongation. It helps in increasing the length of the plant. That's what we know. This is about the primary meristem, right, bache? And you know that in the primary meristem, we have the example of apical meristem. We have the example of intercalary meristem. We have already covered this part. Now, when you talk about the secondary meristem, it is secondary in origin. Now, what is the meaning of being secondary in origin? Yes. Secondary meristem, as I said, it is secondary in origin. So what is the meaning of being secondary in origin? That your permanent tissue, right, your permanent tissue will undergo de-differentiation. Your permanent tissue will undergo de-differentiation, right? It will be de-differentiated, means cells will regain the ability to divide. Right, cells will regain the ability to divide. So your permanent tissue will be de-differentiated. Its de-differentiation will occur, and we will get the secondary meristem. Okay. Okay, so initially that cell was performing a particular function. Now it has again started the division. Now it is the secondary meristem that we have. So secondary growth is the result of de-differentiation of the permanent tissue. That's the first point. Now when we talk about the secondary meristem, we have the examples, bache, and here in this particular topic we will see that how these secondary meristem will form. So what are the examples that we have? Yes, the first example that we have is of vascular cambium, and specially which cambium it is? It is your interfascicular cambium. Yes, It is your interfascicular cambium. Clear, bache? And then you will talk about the cork cambium. What are we going to discuss here? We are going to talk about the cork cambium here, also known as phellogen, right? And other than that, right, when it comes to the secondary tissues, secondary permanent tissue, then we will discuss secondary phloem, xylem and secondary cortex. But this is the main thing that we need to understand, right? So let's focus on this part. So now you know that in the case of dicot stem, just now we discussed the section of the dicot stem. So I hope you remember. And yes, this secondary growth is in the case of dicot stem. Even in the dicot root, secondary growth is possible, and even in the gymnosperm. So gymnosperms may root and stem, dicot root and dicot stem, they show the secondary growth. Okay, they are going to show the secondary growth. So now here you guys can see that in the case of dicot stem, that's how our vascular bundle is present in the form of rings. And you know that within the vascular bundle, we have intrafascicular cambium. That's what we have. We have the intrafascicular cambium. That's what we have. Yes or no? That's what we have. Intrafascicular cambium is there, yes or no? Intrafascicular cambium is there, yes or no? Yes, we have this. Now, that is why, because this intrafascicular cambium and fascicular cambium, it has formed, right? This cambium has formed during the primary growth. So that is why we even call it as primary lateral meristem, because it is present on the lateral side. So lateral word we are using, and its growth is primary. So it is primary lateral meristem. What is it, bache? It is primary lateral meristem. What is it? It is the primary lateral meristem. Now what is going to happen here? The cells in between these vascular bundle, you know that they are medullary rays, right? They are the medullary rays or the vascular rays. And you know that here the cells are parenchymatous. What type of cells are there, bache? They are parenchymatous cells. What type of cells are there, bache? Parenchymatous cells. Now these cells will again become meristematic. Their dedifferentiation will occur and these cells will become meristematic, and here these cells will start dividing and they will form interfascicular cambium. What will they do? What will they do? What am I saying? That the cells present in between these vascular bundle, these are the medullary rays, yes or no? These are the medullary rays, yes or no? These medullary rays will again, the cells here will be dedifferentiated. The cells here, they will be dedifferentiated, and once they are dedifferentiated, they will again become meristematic. They will again start their division. And what is going to happen here? We will see, yes, what will we see here? We will see the interfascicular cambium. Which cambium will form here? We will see the interfascicular cambium. Just a minute. Okay, see this one I'm talking right now, in between the vascular bundles here. So obviously it is because of the activity of your medullary rays. The cells of your medullary rays will become meristematic. Are you getting it? This one. So can I say that now there will be a complete ring of the vascular cambium? Now after this, can I say that there will be the complete ring? Now there will be the complete ring of the vascular cambium. So right now this is what we are discussing, the formation of the vascular cambium. Clear, bache? Formation of the vascular cambium. Yes or no? Yes or no? Cambial ring will form. Sure? Right? So now when you say vascular cambium, but everything is written in NCERT, so that is why firstly I will explain you this process. Okay, my main focus is to explain this, to make it clear to you all, right? The rest you can read it from NCERT. So first of all, try to understand this. So when it comes to the vascular cambium, so what do we have? we have interfascicular cambium. We have interfascicular cambium, yes or no? And then we have intrafascicular cambium. That's what we have. One is intrafascicular cambium, another one is interfascicular cambium. These are the two words that we have here. So when it is inter is a secondary in origin. When it is inter, it is secondary in origin. When it is intra, it is primary in origin. Just now I told you. Right? So can I say that in dicot stem, yes, in dicot stem, vascular cambium is partly primary, like half of it is primary in origin and partly secondary in origin. Can we say so? That it is partly primary and partly secondary in origin. Can I say so? Yes, can I say so? So this is the formation of the cambial ring. So in dicot stem, it is partly primary, partly secondary in origin. Clear? So that is why in the case of this intrafascicular cambium, also known as fascicular cambium, we also call it as primary lateral meristem. Right? We also call it as primary lateral meristem. Done, bache? Okay. Now the second part of this particular topic is activity of the cambial ring. It is activity of cambial ring or vascular cambium. It is the activity of cambial ring or the vascular cambium. Now, obviously, this ring, now I will only consider the ring. Fine, I will only consider the ring. So, obviously, bache, it is meristematic. So, if it is meristematic, it will do what? It is going to form the cells. If it is meristematic, what will it do? It will form the cells, yes or no? If it is meristematic, it will form the cells, yes or no? Of course. So this cambium, when you talk about this vascular cambium, now you know vascular cambium is having two parts. So when you people talk about this vascular cambium, it will start dividing, right? We basically discuss it in two ways, that some ray initials are there, fusiform initials are there, periclinal division, this and that, but I'm not going to discuss it in this way. We will keep it very simple, right? Just say in the case of vascular cambium, now we divide it into two parts, fusiform initial and the ray initial. Clear, bache? Fusiform initial and the ray initial. Actually, in the vascular cambium cell, what type of cells are there? We discussed two things. Ray initials, the cells which are oval, right? Which are oval, or you can say that spherical. Another type of cell is fusiform initials. They are long, they are pointed, they are more in number. Actually, these are the two type of cells which are making the vascular cambium. These are the two type of cells which are making the vascular cambium, right? So obviously these cells, they divide, just particularly your fusiform initial, right? Your fusiform initial, because they are more in number. So it is nothing, it is just the type of cells which are making the vascular cambium. So I'm repeating this part again. Vascular cambium having intrafascicular and interfascicular cambium. How that interfascicular, inter means in between, present in between two vascular bundle. How that interfascicular cambium will form? It will form by when medullary cells will become meristematic due to dedifferentiation. Now there is the complete ring of the vascular cambium. So what type of cells are there in vascular cambium? Two type of cells are there, ray initial and the fusiform initial. Ray initials and the fusiform initial. So ray initial, they are spherical cells, less in number. Fusiform initial cells are long, pointed, more in number, right? But here, ultimately, both of them, they will divide and they will form different structures. But in NCERT, they have kept it very simple. They have simply used the word vascular cambium and then the activity of vascular cambium. So it is cambium, for sure it is going to divide. So vascular cambium, it is more active towards inside in comparison to It is more active towards inside, towards inner side basically. Now, what is the meaning of this particular statement? It's very simple. Bache, your cambium is going to divide. Fine. Your cambium is going to divide. Your cambium is going to divide. Towards inner side, it will form secondary xylem. Of course, xylem is present towards inner side, so it will form secondary xylem. Towards outer side, it will form secondary phloem. Okay, towards outer side, it will form secondary phloem. Towards inner side, it is forming secondary xylem. Towards outer side, right? Towards outer side, it is forming the secondary phloem. Now, I am saying that it is more active towards inside. It is more active towards inside. So it means you will see more secondary xylem in comparison to the secondary phloem. The formation of secondary xylem is more than that of secondary phloem, yes or no? The formation of secondary xylem is more than the secondary phloem, yes or no? Of course it is. Okay, of course it is. So you will see that more secondary xylem will form. In comparison to it, your secondary phloem will be less. And the reason for it is obviously the growth factors and all they are responsible. Okay? Growth factors and all they are responsible. So any doubt? Any doubt? It's a direct question, bache. Right? It's a direct question. Okay? So secondary xylem is more, secondary phloem is less. So secondary xylem is more, secondary phloem is less. So you can even write it down that secondary xylem is 8 to 10 times more than 8 to 10 times more than the secondary phloem. Are you getting it? It is 8 to 10 times more than the secondary phloem. Clear? Sure? Sure? Yes. So unequal distribution of hormone is responsible for it. Yes or no? Yes or no? Now, see bache, already you know na, primary xylem was present there and the secondary phloem was also there. I hope you know about it. I hope you know about it, that already even the secondary primary xylem was present, even the secondary phloem was there. है ना? So primary xylem, it is present towards this pith region. It is inner to this. Can I say that? Just look at the positions here, bache. Just look at the positions here. Can I say that it is inner to the secondary xylem? Primary xylem is inner to the secondary xylem. Right? Primary xylem is inner to the secondary xylem. Can I say so? Yes or no? Yes or no? Can I say so? Of course. And here you people can see, right, that your secondary phloem is present outside to this. Your primary phloem is present outside to this vascular cambium, outside to this secondary phloem. Right? So you need to remember the position as well. Fine, you need to remember the position as well. Now, obviously, it is exerting a pressure because the growth of secondary xylem is too much. So, of course, it is going to exert a pressure. It is going to exert a pressure on this primary xylem. Yes or no? The secondary xylem, the growth is too much. So, obviously, it is exerting a pressure on the primary xylem as well. It is exerting a pressure on the pith as well. But somehow, your primary xylem will remain intact. Your primary xylem will remain intact, right? It will not get affected that much. Now, obviously, towards inner side, you are increasing the cell number. You are increasing the cell number. You are increasing the cell number. Obviously, even it is going to push, even it is going to exert a pressure over the old formed secondary phloem and primary phloem. Yes or no? Think about it. Just suppose karo in a balloon, you are just blowing the air, there comes a time when it will burst out. Okay. Now you can even take the example of an animal cell. Animal cell is without cell wall. You are putting it in a hypotonic solution. Water is moving in, water is moving in, water is moving in, water is moving in. There comes a time when it will get burst. right? There comes a time when it will get burst. Same as the case here. So obviously secondary xylem, the growth is increasing, the cell number is increasing, the cell number is increasing, the cell number is increasing. It is going to exert a pressure. It will even break the old formed secondary phloem and the primary phloem as well. It is going to crush it. It is going to crush it. Yes or no? Yes or no? Yes or no? है ना? है ना? So that primary phloem will also get crushed. Old formed secondary phloem will also get crushed. And with the help of this diagram, the position of things should be clear to you. So this is the primary xylem. This is the secondary xylem. This green one is the cambium. This one is your secondary phloem. And this one is your primary phloem. I hope positions are clear to you all. Okay, so it will get crushed. It will get crushed. Any doubt? Yes, any doubt? And now, after the formation of this, chalo, let's. Array. So that's how it is present, yes or no? That's how it is present, yes or no? This one is the cambium. Let's consider it as cambium. Secondary phloem. Primary phloem. Secondary xylem. Primary xylem. Primary xylem, right? So this is all the activity of your fusiform initial. Okay, now what is going to happen, bache? Yes, what is going to happen now? Now the ray initials will also divide. You don't focus on ray initials or the fusiform initials. Simply you keep it in your mind that from the secondary xylem and phloem, right? From the secondary xylem and phloem, there will be the formation of, right? There will be the formation of the, the division will also be there, right? You will see some rays. So these are the secondary medullary rays. Okay, from the secondary xylem and phloem, secondary medullary rays or secondary vascular rays will form, right? Secondary medullary rays or secondary vascular rays will form. Is it clear? Yes, is it clear? Done? Any doubt, bache? Just a minute. So secondary medullary rays will also form, and they are what? They are yes, they are responsible for the radial conduction of food. Done, bache. So now let's say compare it with the diagram. This one. So see the completion of this ring, right? And then see the xylem and phloem will be formed, and there comes a time when what is going to happen, bache? These are secondary, right? These are secondary medullary rays will form. Clear, bache? These secondary medullary rays will form. So obviously cell number is increasing. So the girth is increasing, diameter, circumference is increasing. Yes or no? Of course it is increasing. Right? Of course it is increasing. So this is all about the activity of this vascular cambium. Clear, bache? This is all about the activity of this vascular cambium. Now, especially in the temperate regions, right? Do you know temperate regions where seasonal fluctuations are there, where seasons are not uniform throughout the year? Do you know that? In temperate regions, you know it very well that seasonal fluctuations are there. Seasons are not uniform throughout the year. Seasonal fluctuations are there and seasons are not uniform throughout the year. This is what we know. Temperate region, obviously Asia comes under it, and even the North America also comes under it. Right? Temperate regions. I'm not talking about the tropical regions. I'm talking about the temperate regions. So temperate regions में seasonal fluctuations are there. What is there in the temperate region? Seasonal fluctuations are there. So now, bache, when you people talk about the cambium, so the cambium activity changes. Cambium activity changes with the season, right? It changes with season. Like when it is the spring season, so cambium is highly active, right? When it is the spring season, your cambium is highly active. जैसे देखो बच्चे, activity of this cambium is no doubt under the physiological factors are controlling it. No doubt the activity of this cambium is under the control of physiological factors, but environmental factors are also playing the role. Okay, environmental factors are also playing the role. So what is going to happen there, right? Activity changes with season. Cambium activity changes with season. So when it is the spring season, cambium is very much active, and in comparison to the autumn season. Okay, in comparison to the autumn season. So in cambium, in spring season, cambium is more active. So obviously it will form more secondary xylem. Like its activity is more. So obviously more secondary xylem it will form. Yes or no? More secondary xylem it will form. Actually, bache, this secondary xylem is the wood. Like in the case of the dicots, you can take the example of any dicot plant, dicot tree. You know that, right? That thick stem is there. So that woody part is majorly your secondary xylem. What is it? That woody part. Okay, woody part is what? That is your secondary xylem. So here what is going to happen? Your secondary xylem is more secondary xylem will be formed. Cambium is active. In comparison to phloem, secondary phloem, more secondary xylem will be there. But ultimately, if I compare spring season and autumn season, spring season mein cambium is active. Here cambium is not so active. It is not so active. So in comparison to spring season and autumn season, you will have less growth. You will see the less secondary xylem. The formation of less secondary xylem, isn't it? The formation of less secondary xylem will be there. So spring season mein, the wood that will form is known as the spring wood or the early wood. Okay, we will call it as spring wood or the early wood. And in the case of this autumn season, it is going to be the late wood. Fine, it is going to be the late wood. Now, as I'm saying, more and more xylem elements are there. More and more xylary elements are there. So here you will see that tracheids, vessels, right? Their lumens are wider. Lumens are wider here, right? Lumens are wider here. So in such case, the spring wood is having less density because lumens are wide here. Why they are wide? Are relate it na? It's very simple. There is no need to cram it. Guys, there is no need to cram it. Now my cambium is more active. It is forming, it is doing a lot of activity. It is very active. It is very, very, very active. It is adding more secondary xylem. It is adding more secondary phloem in comparison to the autumn season. So obviously it needs more water. It is more active. It needs more water for its activity. Yes or no? It is more active, so it needs more water for its activity, isn't it? Isn't it? So whenever it is adding the xylem elements there, they are going to have the wider lumens so that more water can come in. More water can go up from that part. Okay, so because lumens are wider here, so the density of that wood will be less. Fine, the density of the wood will be less. Clear, bache? Yes or no? Is it clear? So autumn wood and the late wood. Next is the autumn wood and the late wood where vascular cambium is less active. Okay. So here you will see that vessels are what? That vessels have narrow lumen. That vessels have narrow lumen. Yes or no? The lumen is narrower there. Isn't it? The lumen is narrower there. Okay, so density of this wood will be, density is more. Clear? So in the case of, right, in the case of such plants which are growing in the temperate region, right, when you people will take the cross section or transverse section of the stem there, and always, right, so imagine there is a dicot stem which is growing in the temperate region. So if you have to take the section, you will observe its trunk, especially near to the base. So you will, because in one season cambium is more active, in one season it is less active, right? Even the wood density and the color will also be different. Yes or no? Wood density and the color will also be different. So what are you going to observe there in that case? You will observe some rings, right? You will observe some rings, some concentric rings you will observe. What will you observe? You will observe some concentric rings there, right? So these are two rings, okay? These are two rings are showing one complete ear. I told you in temperate region uniform distribution is there. Yes or no? This is what I'm saying. Yes or no? Yes or no? So what will you see? What will you see? You are going to see two concentric rings. You are going to see two concentric rings. Concentric rings means having same center. And these two rings are showing one growth year. Right? Two rings are together showing one growth year, and we use the word annual rings for that. We use the word annual rings for that. And let me tell you students, the spring season may, the spring wood is lighter. Okay, spring wood is lighter in color. It is less dense, so it is lighter in color. And the autumn wood, the late wood will be darker in color here. Okay, it will be darker in color here. Is that clear? Yes or no? Is that clear? So obviously two rings together, they are showing one growth here. It is showing annual ring. Fine. It is showing annual ring. Done? Done? So by counting the number of rings, what is going to happen? We can even check the age of that particular tree. Okay, we can even check the age of that particular tree. And this technique of calculating the age of a tree by counting the annual ring is known as dendrochronology. But this word is not given in NCERT, but yes, it's a previous year question, so you better note it down in your notebook. That is the dendrochronology. What is it? That is the dendrochronology. So what are we doing here, bache? We are just counting the annual rings. What are we doing here? We are just counting the annual rings here. Okay. So two annual rings, two concentric rings, two annual rings, two concentric rings means 1 year. So that's how you can even calculate the age. Okay. You can even calculate the age. Done? Sure. So spring wood, autumn wood, clear. Yes, spring wood, autumn wood, clear. Now you know that this process is not a one-day process. It is going to take years. है ना? This secondary growth is not a one-day process. It is going to take the years. So obviously, when your secondary xylem, but keep it in your mind always that secondary xylem is actually making your wood. है ना? It is the secondary xylem actually which is making that wood part. It is actually the wood. Yes or no? Secondary xylem is actually the wood. So when the older secondary xylem, obviously the older secondary xylem will come in the center, right? Will come in the middle part. So this older secondary xylem will form the wood which is hardwood or also known as duramen. Let me explain how. Just imagine it. Imagine it. Like let's say this is the vascular cambium. This is the vascular cambium. Towards the inner side, let's say this is the inner side. So towards the inner side, there is the formation of secondary xylem. Towards outer side, there is the formation of secondary phloem. So with the time, secondary xylem will form more and more. And the central part, right? Imagine that central part. It is the older secondary xylem, the wood. It will become darker with the time. Do you know that? It will become darker with the time. Right? It will come in the center. It will become darker with time. Why? Because there will be the depositions. Why is it so? Because there will be the depositions of tannins, okay, deposition of renins, tannins, gums, resins, and all of these things will get deposited there in the xylem elements in their lumens, right? All of that will get deposited there because that is the older secondary xylem. So now what will it do? Newer is also getting added. So what will it do? The depositions will be there. So because of that depositions, its color will become darker. So heartwood, heart in Hindi is dil. So heart, HD. You can even remember HD, high definition. So heartwood is known as duramen. Heartwood is known as duramen. Heartwood, I'm repeating this again, guys. Heartwood is known as duramen. So here there is the deposition of the tannins, deposition of the renins, deposition of the gums. Okay, so obviously it will become microbial resistant. Microorganisms will not be able to attack here. And with the time, what is going to happen? You know that in xylem we have the xylem parenchyma. We have the xylem parenchyma. In the xylem, we are also having the xylem parenchyma. Yes or no, students? Be quick. Yes, be quick. Yes, we have the xylem parenchyma. So this xylem parenchyma in this heartwood, it will form some outgrowths. Do you know that? This xylem parenchyma, it will form some outgrowths. So these outgrowths will be there in the lumen. In the lumen of vessels, these outgrowths will come. जैसे xylem, now depositions are there. Xylem parenchyma is showing outgrowths. That outgrowths we are going to see in the lumen of the tertiary elements. So these outgrowths are acting like a plug. We also use the word tyloses for it. Right? These are the outgrowths present in the lumen of the xylem. These are the tyloses. Outgrowths. These are acting like a plug. Right? So obviously they will inhibit the water transportation there. They will not allow the water conduction there. So when it is the heartwood, it will not conduct water. Right? It is not meant for the water conduction, although it's a heart. So plant ke case mein that heart is not conducting anything. Right? It is just a wood, dark in color, having the depositions of the renins and the gums, bache, due to which it will become what? Yes, bache, it will become darker in color, microbial resistant, and it is not going to conduct the water. So it is the central region of the secondary xylem. Clear, bache? It is highly lignified. It is the central region of the secondary xylem. Clear, bache? So these tracheal plugs are present which will block the lumen of the tertiary elements. Done. And in the surrounding, just suppose the xylem, this is the secondary xylem. So this part is there in the center. So in the periphery, what you people will see. In the periphery, what you people see? Right here you will see lighter part, lighter secondary xylem. Actually, it is the sapwood. It is known as alburnum. Right? It is actually meant for the conduction of, the water and minerals. right? It is actually meant for the conduction of the water and minerals. So center mein you will have hardwood, then surrounding mein, periphery mein you will have sapwood, which is lighter in color. There is no deposition, so it is meant for the water conduction. Any doubt? It is meant for the water conduction. Any doubt? Sure? Sure? Clear? That is about the hardwood and the softwood. So this is all about, see, everything is given in NCERT. Okay? Everything is given in NCERT, bache. So obviously I am not going to read it right now. I have already explained it in a very good way, so you guys can read it after the class, and if there will be any doubt, you guys can mention that doubt in the comment section, and obviously I will answer it. Done. So hardwood, sapwood. Hardwood is your duramen, and when it comes to the sapwood, it is known as alburnum. It is known as alburnum. So out of these, this one, it is the sapwood which will conduct water and mineral. So it is meant for the conduction. Okay, it is meant for the conduction. So next is the cork cambium that we need to discuss, right? So now we will talk about the formation of the cork cambium and its activity. Formation of the cork cambium and its activity. That is what we need to discuss. So any doubt, anything that you know about this? Yes, anything that you know about it? Tell me. Tell me. This is important. So, bache, cork cambium, now you know that. Now the secondary phloem is increasing. Secondary xylem is also exerting the pressure. Secondary phloem is also increasing. So obviously they are going to break the cells which are there in surrounding. They are going to break what is there in the surrounding. Let me show you here. What is there in the surrounding? So surrounding mein you have the pericycle cells here, right? You have the endodermis here, basically the starch sheath, because it is the dicot stem, so obviously it is the starch sheath. You are having the cortex here. So ultimately what is happening when all these cells and this tissue is increasing, it is going to exert a pressure here also, and it is going to break it as well. It is going to rupture it as well. So we need to form new cortex. Okay, we need to form new epidermis, because the tissue that is increasing is breaking the outer layer. Okay, it is breaking the outer layer. So what are we going to discuss here? We will talk about the cork cambium. So how this cork cambium will form, bache? The hypodermis, right? The hypodermis will be dedifferentiated. This hypodermis is going to form that. Or simply you can also use the word cortex. Clear, bache? Simply you can also use the word cortex. So cork cambium, also known as phylogen, it is derived from the hypodermis, right? It is derived from the hypodermis. So cork cambium is what? Phylogen. Gen means to generate, to form something, right? Cork cambium, phylogen, to generate, to form something. So cork cambium, it is derived from the dedifferentiation. Always secondary growth is because of dedifferentiation. I told you already. So it is derived from the dedifferentiation of hypodermal cells. So hypodermis is nothing. It is the outermost layer of the cortex. It is the outermost layer of the cortex, right? Is it clear? Is it clear? So obviously the cells will become meristematic and they are also going to form. Let me draw it. Right? They are going to form what? Cork cambium. So now obviously even this cork cambium will divide, bache. Even it will divide. So towards its inner side, it will form secondary cortex. And that secondary cortex is known as, bache, phyloderm. What is it? It is known as phyloderm. So towards inner side, it will form secondary cortex, which is known as phyloderm. Towards outer side, it will form cork, right? Right, it will form cork, and here cells are dead, lignified, suberized as well. right? Suberized as well. So outermost portion is known as cork, and we also use the word phellum for it. So outermost cells, after their formation, they will become what? They will become dead bachcho. Yes, they will become dead bachche, right? They will become suberized, and they are known as cork or phellum. And towards inside, what will you get? You will get the secondary cortex or the phelloderm. So innermost cells are living. So obviously they will be parenchymatous. They will be parenchymatous. And here, outermost side pe, they are suberized, means they are dead, they are water impermeable. So that's how it works. Clear? So cork cambium, the phellogen, outermost side pe phellum, innermost side pe phelloderm. This is what it is doing. Clear bache? Clear bache? Yes or no? Yes or no? So now here, this phellum, this is important, please note down. Phellum, phellogen. So when it is gen, to generate, so it is the cork cambium. And this phelloderm, they are together, they form. These three are together known as periderm. Right? So what is periderm? What is periderm? Phellum plus phellogen plus phelloderm. What is periderm? Phellum plus phellogen plus phelloderm. It is a direct question. Okay, it's a direct question. Done? So what is periderm? These three layers together, phellum, phellogen and phelloderm, they form the periderm. Are you getting it? They form the periderm. Clear bache? Clear? So obviously if cork is suberized, so can I say that it is impervious to water, that it will not allow the water movement? It will not allow the water movement. If I'm saying it is suberized, so can I say that it is impervious to water? Right? It is impervious to water. Yes, of course I can say that. So bache, this cork made up of dead cells, made up of suberized cell, it is also in some trees, na, this cork is having the commercial purpose. This cork is having the commercial purpose as well, like oak tree. Okay, so this is what you need to remember here. So that's why it is adding. Right, that is how the cells will be added here further. Now, what is the important point? Now we need the NCERT. See, this is important. Please focus. As the stem continues to increase in girth, hai na, due to the activity of vascular cambium, outer cortical and epidermal layers, it will get broken. They need to be replaced so that new protective cell layers can be added. Of course, they need to be replaced so that we can add more protective layers. So sooner or later, another meristematic tissue called cork cambium or phellogen develops. So it develops usually in the cortex region, right? So I specified it. Right, so mainly in the cortex region, this cork cambium will develop. Now, mainly it is the hypodermis which will form the cork cambium. Clear, bache? It is actually the hypodermis that will make the cork cambium. So phellogen is what? Bache, once a question was there from this part. So such type of statements, such type of question, such type of lines can come in statement based question. So you should know about it. Like when you talk about the phellogen, it's a couple of layer thick, means it is a multi-layered, right? Please note down. Please make the notes. It is multi-layered. Now, it is made up of narrow, thin-walled and nearly rectangular cell. Obviously, cork cambium is meristematic, so it is going to be thin-walled. Cork is thick-walled. But when it comes to the cork cambium, it is thin-walled because they are meristematic. But what type of shape is there, Ishwat? It is rectangular. Right, it is rectangular. So phellogen cuts off cells on both the sides. Obviously, outer one is the cork or phellum. Inner one is the secondary cortex. Another important statement. Clear? So cork is impervious to water. You know why? Because a suberin is deposited, but your secondary cortex, that is your parenchymatous. So these three together, they are forming the periderm. Any doubt? Any doubt? So obviously when this cork cambium is dividing, pressure builds up on the remaining layers to the phellogen, and ultimately these layers will die and slow off. Clear? Clear, bache? These layers will die and slow off. So now, bache, we have one more term which is non-technical, and it is a question in NEET 2023. Right? It was a question in NEET 2023. So we use the term bark. Right? We use the term bark. So bark is a non-technical term. It's not a technical term. Sometimes some people confuse it. It's a non-technical term. It's a non-technical term. Right? It is not technical, it is non-technical term, right, that refers to all the tissues present outside the vascular cambium, right? Present outside the vascular cambium. So what will be there in the bark? Please make the notes. What will be there in the bark? First of all, bark is a non-technical term. So bark include periderm plus secondary phloem. Periderm plus secondary phloem means it includes that cork cambium, secondary cortex and secondary phloem. Done? That is what bark is. Clear, bache? That is what bark is. So it includes periderm plus secondary phloem, cork cambium, secondary cortex and secondary phloem. So obviously these are the tissues which are outside to the present outside to the vascular cambium. Clear, bache? They are present outside to the vascular cambium. So this is all about the activity of this cork cambium as well. Okay? So cork cambium will remain living and active only for 1 year. So every year new cork cambium is formed. Clear, bache? Every year new cork cambium will form. Fine. Sure. So this cork cambium is active for 1 year only. Every year new cork cambium is formed. Sure. Okay. So next is the lenticel. I hope you know about the lenticels, right? They are the pores. They are the pores which are present on the bark, which are present on the stem basically. So they are meant for the gaseous exchange. Okay, these pores are meant for the gaseous exchange. So see here, barks refer to the number of tissue I told you, periderm and secondary phloem. So it is formed early in the season is early or soft bark. If it forms in the end of the season, it is the hard bark. So now there is one question, name the various kind of cell layers which constitute the bark. So that's your homework, right? You will answer it in the comment section. Okay, so I hope you all have checked this particular video, that how to read the NCERT, right? How to read the NCERT. Here in this video I told you this thing very clearly, that in NCERT sometimes they left some statement, they put the question mark there. Some statements are, you know, they are asking you to answer them. So question can come from that part also. So if you have checked it, very good. Otherwise do check this video. Clear, bache? Chaliye, next. So here you guys can see the location that I have added, that location of some of the primary and secondary tissues, bache. So here you people can see the oldest layer of phloem, means your primary phloem, lies just inside to pericycle, of course. है ना? Your primary phloem lies just inside to the pericycle, isn't it? And your oldest layer of secondary phloem lies just inner to the primary phloem. है ना? It is even inner to the primary phloem. Youngest layer of phloem, that is your secondary phloem, it lies outside the vascular cambium. So youngest layer of xylem or secondary xylem, it lies inside the vascular cambium, and oldest layer lies outside the primary xylem. So you guys can refer it and write. And if you understand this topic properly, then there is no need to cram it, bache. It is the easiest topic, let me tell you. Actually, it is the easiest topic. Done? Actually, it is the easiest topic. So here you people can see this diagram. Okay, this is basically of lenticels. Let me show you. So at certain regions, phellogen means cork cambium, it cuts off parenchymatous cells on the outer side instead of cork cell. This line is very, very, very important. You know that on outer side it is forming the suberized cell. It is forming the suberized cell. It is forming the cork. But here at some places, this cork cambium towards the outer side, it is forming some living cells. Right? This point is very important. But your question can come from this part. Okay, this point is very, very, very important. So these parenchymatous cells, once the word is parenchyma, always keep it in your mind that it will be living. So these parenchymatous cells, they will rupture epidermis. They will form lens-shaped opening called lenticels. So these lenticels, they helpful in gaseous exchange, right, between the outer atmosphere and the internal tissue, and it occurs in most of the woody trees. So look at this diagram, so here you guys can see the epidermal cells will break off, right? Here you have the living cells. They all are living. These are the complementary cell, and it is making the lenticel, right? So lens-like structure will be formed. That's what you need to keep in your mind. Done. And this is the bark. So now when it comes to the secondary growth in roots, even it is very simple. See, you know that it is possible in the case of dicot roots. So when you know the roots are dicot, when you know it is a root, the vascular bundle is radial. Yes or no? That's very easy, bache. The vascular bundle is radial. Exact. Xylem is arranged, right? Xylem is arranged xylem is arranged. How is it arranged? The protoxylem is facing the periphery. Exact arrangement is there. So in between the xylem and phloem, here you will see the parenchymatous cells, and for them we use the word conjunctive tissue. So in the case of secondary growth, right, or especially in the dicot root, right, when you talk about the vascular cambium, it is completely secondary in origin. This vascular cambium is what? It is secondary in origin. And you guys can see here that this conjunctive tissue, these parenchymatous cells, they will be de-differentiated. Okay, they will become actively dividing. They will be de-differentiated. They will become actively dividing. They will be de-differentiated, they will become actively dividing, and here even the pericycle cells also become meristematic. So here, you know, what are you going to see? You will see that vascular cambium that will form is not circular, it's a kind of wavy ring, right? It's a kind of wavy ring. Let me show you the diagram here. So here the vascular cambium will form, but it is not a circular ring, it is wavy, but with time it will become circular, and vascular cambium will have same story here also. Here secondary xylem, here secondary phloem, and rest everything is same, but this is the only part which is different. Okay, it is the only part which is different. So see, in the dicot root, vascular cambium is completely secondary in origin. Very important MCQ, bache. It originates from the tissue located just below the phloem bundles, a portion of pericycle tissue above the protoxylem, forming a complete and continuous wavy ring. So basically this conjunctive tissue, right, this conjunctive tissue present here will become meristematic, and the cells, right, see this portion of pericycle tissue above the protoxylem will become meristematic. So that is why they will form a wavy ring. Array, what type of ring will they form? Okay, wavy ring. And then slowly more cells will be added, it will become circular, and rest all the steps are same like your secondary growth in stem. Okay, so secondary growth, I told you already, it occurs in the stem and roots of the gymnosperms also. Fine, but it is not there in the case of monocots. So that's it. That's all about this particular chapter. So one more chapter down, bache. See, cell cycle done, biomolecule done, reproduction, human reproduction done, reproductive health done, anatomy done, right? And other chapters are also there, na? I'm not able to recall. Your living word done, biological classification, animal kingdom, plant kingdom done. So see, structural organization in animals, tissue part done. So now you tell me, right, I have finished that number of chapters. Now you tell me, what do you know from your syllabus? And guys, still you have the time. Let me tell you this thing very honestly, still you people have the time, right? Please utilize it properly. If there is any problem that you guys are facing, please put that in the comment section. I always check the comments. I will make the videos on that, and I will make sure that you score well in your NEET examination. But before that, you have to decide what are the minimum marks that you want to score there in the NEET examination, and you really need to work hard for that, bache. If you want to be a doctor, right, your NEET examination is not difficult, but the syllabus is vast. It is memory based. Why? Because they are training you, right? They are trying to train you for your MBBS syllabus. You need to know about the human body. You need to remember everything about the human body. Then only you can give life to someone. This journey is never going to be easy. It is going to, it will demand everything from you. You have to step out of your comfort zone. Otherwise, you will not be able to crack it. You know that every year number of aspirants, they are increasing. 22 lakh aspirants were there. Near about 22 lakh aspirants were there in the last year. And this time it will go above 23 lakhs. So please start studying properly, guys. And that's why we are launching batches like this, bache. That's why, right? That's why we are launching such small courses which can help you to, you know, master a particular subject. And one of it is, you know, that chemistry mind map by HSP sir. And you know that sir is brilliant. Okay, so be the part of this batch, and please be regular in this batch. You are, okay, you are going to do wonders there, trust me. And I think maybe there is some issue with the link is not working well. Okay, so don't worry, I will talk to my team. It will be okay as well. You please join this batch. Okay, you please join this batch. You can see his videos, right? His mind maps, they are absolutely amazing. Clear? Okay, so take care everyone. You know what you have to do in the comment section. It will hardly take your 10 seconds. Please let me know you like the session or not, and please, the homework question you need to answer in the comment section. So take care. Thank you.